Hello and welcome to this webinar titled Implementation Strategies and Challenges for Single-Use Technologies at Commercial Scale, hosted by Biopharma Asia magazine and presented by Adam Goldstein at Roche, Genentech and Jacob McNeil at Thermo Fisher Scientific. My name is Orsolya Balog and I will be the moderator. Before we begin, I would just like to remind our viewers that there will be a live Q&A session at the end of the presentation. Audience members can send their questions at any time during the presentation via the Questions tab located at the right-hand side of your screen, and we will go through these at the end of the webinar. Now, please allow me to introduce our first speaker, Adam Goldstein. Adam is currently the Roche Genentech Global Lead for Single-Use Technologies. His current function is to lead and develop strategies for single-use technologies and aid in the tech transfer of new molecules and process engineering changes within the manufacturing sciences areas for development and commercial operations. In recent years, Adam has been the startup lead for both clinical and commercial operations leadership for some of the largest commercial manufacturing facilities in the US, which include 5 significant biotech facility startups, including Genentech, Amgen, Biogen, Baxter, and Genvac facilities. I will now pass it over to Adam Goldstein to start the webinar. Welcome, Adam.
Well, thanks so much for the invitation and the introduction. Look forward to today's discussion. Right, let me move to the next slide. Today I'm going to be discussing with you the challenges and some strategies for single-use implementation. The agenda for today, a little bit of background about single-use technologies, some of the key challenges for disposables, how do we manage the materials? They have a very large, complex supply chain. So what's involved in that? What are some of the challenges? Some of the strategies as well around the management of these materials in construction, and a couple of examples of where we commonly use these SUT operations now in GMP operations. A little background. Single use, as you may know, if you've been looking at the single use advantages and innovation, are rapidly evolving. And these evolving technologies are moving rapidly from the clinical phase into the commercial manufacturing phase for their use. The real driver for these single-use bioprocessing technologies is really cleaning reduction, ensuring sterilization, and reducing the amount of maintenance that you have to perform on steel-based reactor and other downstream systems. The technology is being used now in a broad range of applications. We already know that we use them quite often, even though they may not be looked at as single-use technologies. We're already using filters every day in our common bioprocessing processes in biotech and pharma. They are indeed single-use operations in most cases. There's mixing applications which are disposable, and quite a few purification upstream portions that are disposable. But the use of disposables are rapidly expanding, especially in areas where we want to move into third world markets or rapidly be able to have a flexible facility that's in a modular format that can easily be templated across many different areas or markets. So a little bit about single-use technologies. They're not new. So that's really, it may be new to CHO and bacterial expression system manufacturing, but they're not new. We've used single-use systems quite robustly and often in our daily lives, and just a couple of examples of those are these examples here with blood bags. Baxter was an early innovator. You go into the hospital, you have a loved one or family member in the hospital. And I won't discuss this heavily, but the scrutiny that we put on single-use technologies now in biotech is quite different than the scrutiny that was put on blood bags, plasma bags, where you stick that needle directly into your vein. So that's something to think about. We also, and we'll discuss some of those regulatory discussions briefly. That's not the point of this talk, but briefly throughout this discussion. So we see that they've been used quite often. We trust these disposable plastic applications with our children. We don't think second thing about doing inhalation, which has another route of administration, another level of scrutiny, or drinking from these single-use containers all the time. So I know there is another side of single-use technologies, and if you've been watching the news recently, you see that in the ocean, but that's not the focus of our discussion today. So the focus is really on implementation of single-use technology. So the point here is it's not new to us as a user, but it's a little bit more new to us in the biotech industry. So let's understand some of these single-use applications a little better. Here's an example from one of our websites, internally Genentech Roche, that we use to organize our single-use technologies, and you can see it's broken down by single-use spinners, media buffer hold, bioreactors, connectors, tubing, mixers, etc. And then we also have very detailed organization of the studies supporting the use of each of these single-use components. In this case, this is a snapshot from our website showing some background on tubing, and if you click on it further, it goes into some of the studies and the rationale and validations of the use of these within our company. So understanding the components and how they may or may not affect our product or patients is important. And all those technologies are great. There was some really cool innovation coming out. It's evolving more rapidly than I've seen it in the last 20 years. If you go to a trade show, you'll see more and more new technologies coming out. But getting these technologies into your company, it may not be as simple as you might think, and it really takes a coordinated strategic approach across your company to decide how are you going to implement these technologies in a coordinated fashion so that you don't have 35 different vendors with 35 different supply chains and a number of vendor-initiated changes creating havoc for you to manage within your company, especially in a larger company where you're matrixed heavily with these single-use technologies across organizations. So one way that we believe, one strategy for us that makes it much more lean for us is having a dedicated team of experts who are really focusing on how these technologies interact with the molecules, what are some of the benefits that are derived in a common fashion that we can share across our different manufacturing units and development units. What's a regulatory strategy when you're bringing these things on from early stage development through commercial? And one area that's easily overlooked is shipping and packaging, and we've put a lot more effort recently into shipping and packaging. So, for example, a bulk freeze system with a single-use technology bag, while it might seem intuitive to think about extractable, leachables, bioburden, endotoxin, stability, the protein, all of those are very important factors that we commonly discuss at conferences. But one thing we don't spend a whole lot of time discussing, or at least from what I've seen, is really about the external shipper, the packaging, the temperatures, the glass transition state of the plastics. And those are extremely critical portions that the providing company or vendor may not always have a lot of experience with. And then, of course, a large driver for single-use technology is finance. So how do you justify return on investment, and specifically the procurement team having a strong strategy working with the technical team on how to onboard these materials. So these are key pillars, these 4 key pillars towards an example for us, our strategy on how to manage single-use technologies. So where's the industry really headed? So I tried to compile here 4 main topics here, specifically around their applications, their use in modular facilities, their ability to help us deliver at lower cost per gram, and really allow for us to, and this is related to the modular facility above, really to allow for a more rapid plant startup. And so a little bit about each one of these very quickly. I won't read all of this. Is we know that the single-use applications are being heavily used in media prep and buffer prep and cell culture, but there's also more awareness by the regulatory agencies in what we're doing, how we're doing it, and what the rationalization is for how we use single-use technology. So just a brief comment there on that is understanding who's getting what warning letters, what the expectations are, what the risk-based approaches are, and some of the PDA white papers, for example, on implementation strategies and best practices are very important to understand for implementation. Modular facilities, I'm going to group that with the more rapid plant startup discussion. Having the ability to modularize our unit operations, template them, and then move them from one tech transfer to another tech transfer site allows for greater flexibility, but also greater speed for startup. And I'll discuss that in a few slides coming up soon. If you're able to do this in a well-controlled manner, well-characterized manner, then you should be able to generate lower cost per gram for your manufacturing. That's not always true. And I'll give one good example. If you, and I'm going to use a bad example, but for a biopharmaceutical plant, if you are a large tire company, you're making one type of tire, and you're making just tons and tons and tons of units, SKUs of those tires, and selling them at a heavy level, it may make sense to have, in the case when you take it over to manufacturing, to have a single-use plant. However, if you're going to make winter tires, speed tires, and change over manufacturing plan multiple times to have different types of tires being sold, in that case for a manufacturing plant for tires, then conversely, moving into a biomanufacturing plant, if you're going to do multiple introductions of many different types of new product introduction, then it may not make a whole lot of sense to have one very large stainless steel manufacturing plant. It just doesn't make financial sense. And in many cases, if you're doing smaller volumes and more rapid turns of molecules, single use is very adept and gives a lower cost per gram if you do that analysis. We won't go into that heavily in this discussion. You can look up some of the biopharm publications, and they go into that more deeply. We will touch on it, though. All right, given all that, what are some of the key challenges? Some of the key challenges that we as an industry deal with when onboarding single-use technologies are listed here. So one of the ones that are being looked at much more closely now is around particle risk. So a film that you qualified, you decide to use as a well-characterized film from vendor A that you like to use in cell culture, buffer prep, and media prep, or maybe even pool hold, may or may not be the appropriate film for a final dosage container bag where you're filling vials in the final vial. And understanding how that film is extruded, the N-1 vendor that supplies it to the vendor that you buy that film from, because remember the vendor that you buy your gamma radiated annealed bags from are not, in most cases, the extruder. And so understanding the N-1 vendor process and what level they're extruding the film at is very important and can be very important if you are using it in a very low classification where you need no particulates. Along with that mindset is additives. And I think there's been a lot of talks on this. I won't talk a whole lot today about extractable leachables because I'm sure that many of you have been to many of those talks. If you haven't, we won't go into it deeply today, but it is real. We have learned a lot in the industry as single-use applications are released in new methods and new modes, and they do contain polymers. They are polymers. They have stabilizers. They have antioxidants. In many cases are powder bags with anti-static agents on them, and fully understanding what those ingredients are and whether they mobilize or not into your powders or your liquid stream could have some effect on both your risk assessments and your actual product or patient impact. So really, and that goes back to understanding the extrusion process and where the bags are coming from. Why are these things there? Well, depending on the plastics, where they're extruded or where they're formed parts, they do have release agents that allow them not to stick or they'll roll on the rollers. And so those release agents, as part of, for example, the BPOG protocol that tests for these things, should be looked at closely by your company, and those standard BPOG packages can be requested from the vendor, and you need to have the right expertise, the subject matter expert, to review those reports and understand how a health-based safety assessment can be performed. Leachable is a little bit different. Leachables are actually looking at the final product. Extractable can come from the BPOG material that you get from the vendor, and we'll basically go over what could potentially be there. And leachables are usually done post-Ufdf, final fill products, where you need to get closer to the vial and understand what the total contribution of any leachables to the final product could be. And that's not usually a paper exercise. So how do you manage, how do you understand, how do you get at this information? In many ways, you're required or may be required to do supplier audits. So understanding the raw materials, their components in detail, what type of manufacturing controls the vendor that you purchase your bags from, or their M-1 vendor, or even their M-1 vendor. As we know, there's tubing, there's sanitary connectors, there's extruded rolled bag film, and so in many cases there could be, in that example, at least 3 N-X vendors. So understanding what the manufacturing controls are with your vendor, and does that company have controls to their M-1 vendors. Those are usually dictated in a quality system and quality agreements. So understanding what their quality system is and what you have for your quality agreements with them to manage those risks and those knowns and unknown risks is very important. So again, I mentioned this, identifying your vendors and their backup vendors, and understanding when there would be a change, and would you know if there's a change. And so that's the last point here. So we manage, unfortunately, a reasonably high amount of vendor-initiated changes, and that's what a VIC means. Managing the VICs can be burdensome, especially if you don't have a good quality system and agreement that says, we expect you to let us know if the material changes by X, Y, or Z, and the formulation must be adhered to within a certain specification. All right. Some other critical aspects, packaging. And when I say packaging here, I'm talking about the packaging when the bag or tubing set comes into you. So we've found, unfortunately, that depending on how the vendor folds the bag, whether there's bubble wrap over the tubing connectors or the clamps, that the packaging is a key part to having an integral leak-free system, and the way that it's packaged and delivered and received from both the vendor and then internal to your warehouse is critical. Glass transition issues is really speaking about cold temperature use, cold room, or even freezer uses for bulk freeze, for example. Understanding, are you putting bulk in PETG bottles that are not really compliant at -70? It's a common practice in the industry, but the PETG is really not designed to go at -70 freezers. So we're using the right MOCs, the right material construction, and the right temperatures that they're intended to be used at. And again, we talked about using one film from upstream and downstream. Maybe that one film isn't ideal for freezing. You need a different type of film or different type of bag. Understanding your shipper, your custody. If you do ship something liquid or frozen, you may understand what the shipping container should be at, what temperature should be at, and what your protocol says it should be at. Does it stop halfway from the United States and France, and UPS throws a bunch of dry ice on the top because they think they're doing you a favor and helping you? And that goes below the glass transition state because you have phase material in there that keeps the shipper at -40, and now it's at -70 in the bag or tubings break. Something that's probably happened to us in the past. So, do you understand the custody of your shippers and your single-use materials along your shipping path? What other unknown challenges are there? Such as, and this should be indented, aggregation, precipitation. What's generating those events? Is it well characterized? Is it the stability of protein, or is it an interaction with a slip agent, a post-gamma irradiation, free radical, or something to do with pH shift because the bag does not have a good vapor barrier, and, for example, carbon dioxide from the dry ice in the shipper migrates through the bag film into the material and then shifts the pH, 3-4 units, and causes a precipitation event. So all of these things are real. We've seen all these things. So food for thought. Cell culture testing. So we all have heard a lot about this. I guess my message to you would be you can do extractable leachable, but that might not tell the entire story. And a good example of that would be you get a BPROG protocol. I think they're very important. I think we need them. They're critical to our industry, and they provide some standardization for comparing films and knowing where to use them and how to write up dossiers and defend the use of them in our products. However, we have found where there were protectable peaks found on HPLC or mass spec that shows something that's well within the toxicological level of concern, so you're fine per the extractable leachable testing. However, there's an actual true biological effect, and the cells stop growing. So you can have the graphs and you can have the BPOG data, but in some cases you may need to do biological testing, and not just rely on paper exercises. The last point right here I'll just talk about is training, handling. So let's assume that the packages come in unbroken, they've been packaged right by the vendor, and your warehouse is using sharp box knives, and you know the rest of the story there, and you cut through the box, you cut through the bag, and you don't notice it until you put your expensive protein pool on it, it leaks on the floor. That's a problem. Other problems that I've heard about in the industry and seen and witnessed is, $12,000 single-use bioreactor bags being handled by technicians. They take them out of the box, they drag them across the clean room floor. They're expensive, and the clean room floors are brand new, and they have very sharp edges so that people don't slip when they're wet, and you get micro scratches or leaks by tugging or moving the plastics across, sharp edges or floors. So there's many critical aspects and challenges when working with single-use technologies that you need to take account into. So it may sound like I'm painting a dark cloud over this. I'm not. These are just areas to watch out for. If you're successful with most of those areas, you have good training and good characterization, understanding of your single-use materials. You also need to understand the management of your supply chain with single-use materials. And what I mean by that is we really don't want to play a game like this, and I'll explain this picture here. If you're joining from the United States, you probably know this is a pop-up game. It's called Whack-A-Mole, and your job is on this game is to, wherever a mole steps up out of the ground, you're going to hit it as quick as you can. You never know where it's going to come up from. We certainly do not want to manage our single-use technologies this way. We want to be planned, predictable, understand what's coming into us, and not constantly looking for the next mole that's going to pop up out of the ground. So how do we do that? The way that we do that is really having a good understanding of what the complexity of the materials are. Do we have a really good, strong understanding of the C of A, what the raw material attributes are, and does your quality team and procurement team have a good plan for maintaining consistency both between the vendors and between your plants? So if you're a company like ours that has 6 drug substance sites and 4 drug product sites that's international, and you have vendor A, B, and C across the sites, is it well characterized where you're using the materials, The C of A is shared across all the sites and on the same protocols so that the level of differentiation across the sites is minimized. And this is just a good example here. It's actual true data of the number of increasing materials across our sites. And as we increase the number of materials across our sites, the level of support that comes with it, unfortunately, while not exactly linear, will follow. And so here you see an example of the blue, which are vendor-initiated changes, which we have some control over. So negotiations with your vendor, you should be able to let them know at what level you will accept changes, how many days that you would like to have prior to getting the change, what you consider a change. Still, it's still initiated by the vendor, not by yourself. And then discrepancy management ideally would be zero, but those are discrepancies, whether they're internal from operator generated SOP violations or from the vendor where there's leaks, et cetera. So as you increase your level of single use across your manufacturing plants, you do have to plan for some level of support from manufacturing sciences group and quality to support that level of increase of MOC use. Associated work comes with increasing use of single-use technology. So you may reduce your PMs, you may reduce your passivizations, you may reduce your cleanings and your changeover time, but there's other work that comes with single-use technologies. So how do we manage that? Here's an example. This may not be right for your company, but this is an example of how we currently manage our governance and alignment. We have a single-use category core team. I'll discuss that in just a minute. And then we have three main pillars underneath it. We have our single-use quality team, our single-use procurement team, and then our single-use tech team. So let's start with quality first. So we have our quality teams, and they're really responsible for a vendor-initiated change process, managing relationships with the vendors with regards to quality supply agreements, audits, specifications. Our procurement group, global procurement group, works with the local buyers and local category teams and makes sure that those items which we specify over here on the technical side are adhered to. Any new introductions from the local site are then referred to our technical team, reviewed, and then we have a plan for implementation of those SKUs in a controlled manner. And that way we're not playing the whack-a-mole game across the sites trying to support the different sites and level of implementation. And they are responsible for our supplier relationship management. And so they have quarterly meetings with the suppliers and manage the relationship with the suppliers. The technical team is multifaceted. So we have our validation team, which has a strategy how we do a common family approach for single-use mixers. So is there a worst-case mixer, worst-case size, worst-case dirty hold, how long is the gamma radiation good for, with or without media in it, et cetera. So they generate our standard platforms for validation, and I'm going backwards on purpose here. Innovation, so new innovative products come in through our process development team. And so those new spinners, those new perfusion, those new films, those new candidates that would be truly innovative would be looked at by our development team and then referred on a monthly basis to our technical team, which has all the rest of these team leads represented on it. We have our DS, or drug substance, and our drug product teams, and those teams are more commercial-based. Our manufacturing scientists are on that team, and they're comparing and contrasting what materials are used at the sites, and if there's new introductions, they share the validations and the manufacturing protocols for those single-use technologies. And then we have one last team, which is my personal favorite. I spend most of my time with this team, is the bulk freeze team. And the bulk freeze team is an innovative team. It could be in the development team, but it has a large enough scope. It's looking strictly at just bulk freeze applications and developing a large-scale application to replace cryovessels. So these teams meet monthly and discuss what's going well and how to best organize and make recommendations to the core team. The core team is where we generate the standards with the quality and the procurement partners and make those recommendations to the technical and operational leaders for the sites. This is our Drug Substance Technology Council, our Drug Product Technology Council, our Material Review Board, which is where new materials get approved, and our Material Core Team. And I'm currently the leader for the core and the single-use tech team. So that's a mouthful. That takes energy, that takes organization, but we found that there's a strong payback from having that level of communication amongst the subject matter experts on those teams. So what am I saying there? An optimized management team, understanding the use and single use, the selection, the qualification, and the implementation through some dedicated team of experts is critical. So really organizing what you're doing, where you're doing it, how you're doing it amongst the sites. Having a governance structure I just went over with you to assure your supply and that you can standardize those offerings so you're not playing whack-a-mole game. It may seem really, the beauty of single use is that it's innovative and it's flexible. The double-edged sword to single use is that it's innovative and it's flexible, and that many people from the local sites can specify new unit ops, and with that comes support you may not be ready to support and discrepancies. So managing the change is clearly needed. Understand, these teams also look at the ROA and PV where used in the plants. Another key for us, and I hit a little bit on it, was early adoption. So our development team, that PTD team, really takes a look at what would make the most sense to move into pilot and clinical, and focuses on those films and those new technologies to implement in clinical. One of the teams that is cross-functional, is focused, and they work with all of the teams, and it's not shown on the org chart, is actually dedicated just to material construction qualification and assessment. So they look at generating a risk assessments across the site, so we understand where we're using materials and what the risk of using them are and what data we have support. The team also generated a Ufdf clearance report, which has been published in the industry. Kate Lee, if you're interested, you can look up that publication, showed the level of small molecule clearance for extractables and potential leachables in product streams. And then that helps generate our risk assessment on a risk for each of the unit operations used before Ufdf. And then this team also looks at health-based safety requirements and any other procedures that we need to harmonize our procedures and our use of our SUT technologies. So this is a key, very key to moving into commercial operations and providing regulatory agency updates and filings. One way that that team stays relevant is that the team members have authors and team members for sitting on ISPE, BPSA. Many of them are leaders on BPERG work streams, and also several of them sit as ASTM standard team members. And so they are really taking a footprint and a look of the industry, understanding what the industry expectations are, what the regulatory health agencies' expectations are, and translating them back into our company in the manner of what are toxicological approaches and how do we capture those in risk assessments and procedures. And that way we're really ensuring that we are in line at Roche Genentech with industry and what the expectations are. And conversely, I truly believe that some of the members on this team are helping drive the guidelines through membership, sitting on industry, and helping ensure compliance of what we do is pushed out into industry and to the regulatory health authorities as well. So let's get to a couple of examples of single-use operations. So we talked about leaks, we talked about risk, and some of the other areas. This is just an example where we're showing a pressure decay test, and there's a reason I'm not calling it an integrity test. We shy away from necessarily calling these an integrity test unless we're near final container closure. We're really calling it, a gross leak test. And so before you throw an expensive pool of protein or a large batch of media into a single-use mixer or to a pool hole bag, we've developed with these Genentech single-use sensors on here a way to do a gross leak test pressure sensor. It's quite nice, it's simple, and it just tells us when the bag's in place and it's inflated, do we have any gross leaks or not. So you're not required to do it, but it may be worth your time to do it before you have a catastrophic loss of pool on the ground. Here's just another look at that. It's an example of how we inflate the bag and do the initial stabilization for 5 minutes and then reinflate and look for the passing criteria. Pretty simple, quite nice. One of our actual manufacturing scientists designed this test, and now it's a standard internally for us. So let's discuss real quickly some strategies for management of some of your MOCs as well. So how do we adopt these? How do you really manage the influx of these technologies? And there has been a perceived conflict, not just within our company, but within a lot of companies when I go to conferences and speak with them, that there's a conflict between moving new innovative technologies into the process and then creating delays for launching of a molecule. And so we get that. There is a balance. And many people say, not with my molecule, my timeline's too tight, please move that stuff away from me, I don't want to risk it. But understanding those single-use technologies early in development phase will really help bake that tech transfer in if you do it right early. For example, traditionally, for larger companies, single-use implementation has gone against process development norms. So we really have been swimming against the stream. And so you have a manufacturing plant, it's at the, like, say, 6 by 20,000 liter scale, and you've got robust, big chromatography skids, big stainless steel plant, and you want to put buffer bags in, you want to put a single-use bioreactor as a seed train expansion or a pre-packed column, let's say. You're really entering that here at the commercial stage. You're really swimming against a very hard stream to swim against. You got to update your PASs, regulatory health reviews, SQM, all your certs and specs. This is a difficult way to really introduce single-use technologies. It might make more sense to implement them early. So early on, you do early implementation using a common film, a common type of technology, a well-characterized film, and really template your process development stages. Allows for more flexibility once you have good characterization around the MOC. The operators, technicians, development scientists know how to then transfer that to the clinical sites. And those clinical sites use the same type of MOC. They have the same history for the VIX and the characterization of the films and the challenges for the films so that then they can adopt them in commercial. So this is a much better way to swim with the river than against it. An example of that would look like something like this, and there's no endorsement of this particular bioreactor. It's just an example. It's just a picture. It can be Accelerax, or it could be Sartorius, or it could be a millipore. This is just a nice picture of a thermoreactor. The example would be what I just went over. Would be if you're using this 200-liter sub single-use bioreactor in your pilot plant, and you understand the tubing sets, you understand the connectings, you understand the automation, you understand the leachables, and it's well characterized what grows, what doesn't grow in it, and what type of hassles or advantages that you have with this unit operation. Moving it into clinical should be quite simple. The tech transfer should be pretty seamless, especially if you're working on the same automation system. And so in theory, you could even have cross-trained development scientists and or technicians working on the same processes because you have the same MOCs. So if you do something like this and you have good experience as you scale up in your late phases and you have the same MOCs, taking something in the future from the pilot stage to the commercial stage at multiple scale out instead of scale up creates much more rapid tech transfer opportunities with you for your company with less delays. So that's the paradigm that we're looking at. I've seen other companies look at this paradigm. It makes a lot of sense. And you have future operations with your development well baked in and the materials well characterized. And I think that's my last slide, so I'll just spend a few minutes acknowledging the folks on our team, the large organization that we have that manages these single-use technologies in global manufacturing sciences, process development, our supplier quality lead, corporate engineering, and our procurement teams. All right. Thanks so much. I'll turn it back over. Any questions?
Thank you so much, Adam. Now again, I would like to remind our viewers that there will be a live questions and answer session at the end of the presentation, and that audience members can send in their questions at any time during the presentation via the questions tab located at the right-hand side of your screen, and we will go through these at the end of the presentation. Thank you so much, Adam. Now, please allow me to introduce our second speaker, Jacob McNeil. Jacob currently serves as Senior Manager, Integrated Solutions, Single-Use Technologies at Thermo Fisher Scientific in Logan, Utah. He has a biologics background and has spent the last 19 years in this industry in various roles, such as operations management, setting up single-use manufacturing sites, business market development for customer facility expansion projects, Senior Product Manager of New Product Development, and currently Jacob manages a team of project managers and engineers for customer equipment projects, which includes cell culture, bioreactors, bacteria fermenters, mixers, and controls. I will now pass it over to Jacob McNeil to start the webinar. Welcome, Jacob.
Yeah, thank you, Ursula. And thank you, Adam, for spending the time to go through those challenges of single use. I think it was a great discussion, and there was good points to be made from an end user standpoint on qualifications and handling of the single-use products. So I appreciate that overview and discussion. So from an executive summary standpoint, what we want to go through and discuss really is designing, the facility with single use in mind. Of course, we've seen a huge market uptake in regards to, the scale up into the single use products for the last decade. It's a platform that's growing, flexible, easy to use, allows customers to reduce, facility footprint by a substantial amount. So single use is something that is very common, growing in the markets, and there are challenges with single use in regards to the scale up, choosing the right equipment, choosing the right automation structure, and then of course the right product contact materials and standardizing on those SKUs. In addition, some of the points that I want to talk about is really the process of selecting the right suppliers that best suits and meets your application from an equipment standpoint. Also, the goal should be really looking at productivity and product yields when you're looking at those various pieces of equipment to fill in your facility, and how that really, overlays in with the use of single-use products and how they play a role with one another. And then also touch on, how suppliers manage their quality control process within the manufacturing of these single-use components that are used in the biopharm industry. So first, from a supplier perspective, what's really important from a end user standpoint, I think Adam touched on this, is really planning with the right individuals in place to vet out these various suppliers for best-in-class products. There is a wide range of equipment and materials that are used. So putting together a team, and it's key to plan on that selection process. So, typically what customers do is they will review the various offerings out in the market in terms of equipment. They'll look at the specs, decide on what is valid for their process needs. From there, as an end user, it's important to go through this vetting process, supply those different suppliers with an RFI or request for information, a standard form that can go out to the customers for the various process needs that you do have. Those suppliers will come back to you with their capabilities based on, your requirements. They'll look at quality systems that are in place, innovation pipeline, and really the overall business model. Are they a cell culture company? Are they a media company? Are they a filtration company? Do they have, the ability to provide automation platforms and so forth? Once the RFI process is complete, then again, it's critical that you meet up with those suppliers, really give them an understanding of what your goals are as far as your implementation needs, process optimization, review their quality systems that they have in place, supply chain, are they the manufacturer of the product, or, again, to Adam's point, is there N1 -1 suppliers that are out there that they use for various components within their manufacturing process. Once that's completed, with that detailed discussion and visit from the customer, there's an understanding of what the needs are in regards to the RFP, the user requirement specifications. That discussion goes a long ways. When the URS does come in, there's common knowledge of what those needs are in the process. And then some time for some Q&A to go back and forth to finalize that RFP to ensure that everything's vetted out and straightforward as far as the design of the equipment and the bioprocess containers. From there, we do have a project team that we implement, design engineers, polymer engineers, product engineers. We do assign a project manager to those various projects. Likewise, it's important that the end user has those same technical experts, on those project calls and meetings to ensure that the, product is delivered to specifications and that we're on time as far as scheduling goes. So going to, the equipment side of things, it's really important that, you don't look at, a supplier that, one supplier that looks at having all of the process steps in place as far as equipment, really choose the best-in-class product that suits your needs for that specific application. From there, you'll have a better result in regards to, product yields, productivity, and so forth. So most vendors do have different areas of expertise that you can look into and really get that best-in-class product to ensure that those right design elements are in your process. From an automation perspective, and just in brief, look at the automation platform that you're looking to tie in with your equipment for various processes. So that automation platform, you may have a new facility that needs the MES layer of automation, obviously, which provides a complete process historian, maintenance schedule, operator training, and security to manage that complete process. You may already have an MES system, so from there we can look at unit operation level automation, whether it be a controlling a bioreactor or mixer or so forth, and tie that back into your MES system through open connections or OPC. The one thing from an automation standpoint, I think where you can reduce the number of SKUs that you have in your facility is really looking at the sensors, the transmitters, the pumps, and so forth that are being used across the upstream and downstream process, and choose those components that can be used in those areas. That will help limit those SKU numbers and cost in manufacturing. So again, let's see what's back up here. So again, choose the best, equipment for your process. The automation layer is something that can be added to those various pieces of equipment. This is an example of how these equipments can be tied in through open connection to the overarching MES system or SCADA system that you have in your facility. Choose the best product that meets that process, and then from there we can look at tying that in. In addition, you can also look at a similar design language. So depending on the application, there's an overlay of a software that can be added to each piece of equipment that will allow you to have the same user interface and the same experience across the various platforms within your process. In terms of film structure, this is where it's very critical in controlling the process. Now that we understand the automation layer, the equipment that's being chosen, we transition into the contact material. On the consumable, film choice is one of the most important components within the bioprocess container. This is where your product will spend the most resident time. So it's important that that's very well characterized. The film layer from the supplier standpoint should be the most scrutinized and studied component within the bioprocess container. So from there, mechanical and physical testing is completed on that product. Some of those things that are reviewed are through FTIR analysis for film layer material consistency. So you may have a co-extruded film that has 5 layers. From there, there's analyses that are done to make sure that the layer thicknesses and resin composition is consistent throughout the material. Through the validation, also there's seal strength and process controls which are in place, which includes time, pressure, and temperature on the sealing piece of it. From there, we go into Instron testing that allows you to do a pull test on the bioprocess container to show the seam strength and to ensure the bag is completely integral. On each lot of bag, we also go into a burst test, which is in place from a lot to lot and batch testing. So we'll go through and do burst testing as well. And last, every piece of consumable that you do purchase goes through 100% inspection to ensure consistency and robustness of the consumable. So we talked about the physical and mechanical. There's also, industry standards in regards to the biologics testing, the L&E that is completed. You'll want to review with your supplier how often they do this testing. Thermo Fisher completes this testing on a quarterly basis to ensure that the resins, slip agents with our manufacturers are consistent and are in check. We go all the way down to the supplier level of quality, which means we'll go to our resin suppliers, make sure that those resins and slip agents and the manufacturing process is consistent before that film is brought in and manufactured here in Logan. So again, there will be, of course, from a customer standpoint, additional L&E testing and biologics testing that will be required on that product from both a film layer and a component layer activity. To make sure that the suppliers are consistent with their testing, the one thing that, Thermo Fisher has adopted, and I believe a lot of other suppliers have adopted, is the BPOG process. and standards to ensure that the testing is complete. You know, this is a way for you to review and ask your suppliers for that report on your products to ensure the testing is consistent across the various suppliers of film platforms. So once we've you know, selected the film that's going to be used within the process, there's of course the secondary layer of product contact material, such as tubing, straight connectors, sterile and non-sterile quick connects, tri-clamps, and filters. Again, the point is really to design the bioprocess container to your application for the highest productivity. You may start out with your most complex design, you know, such as your bioreactors or mixers. From there, we can look at the various designs that you have throughout your facility, whether it be a hold container, a tubing set, the bioreactor level, mixer level. Take a holistic approach on your design. From there, we can narrow down the number of SKUs that are used within your facility and the type of connectors and components that you use. So this is where it becomes a little bit more challenging, as you know, Adam has alluded to. There's a number of components out in the market. There's a lot of choice in regards to the connection types, the filters, tubing, and so forth. But depending on your application, whether it's a high pressure application, pumping application, there are certain types of tubing that you would use for those various applications. But again, providing the best-in-class products and tubing and connectors is the most important. What we're seeing in the market is there's continued development of these various components. So as far as the SKU creation of components, it's going to continue to rise, and we don't see this behavior changing in the market. You know, however, the supplier, we have the ability to provide you with information of what the highest use components there are in the industry and what those standards are, to where we can narrow down your options and choice of components that are being used on the design. Key components such as filters, you might want to look at second sourcing, having a qualified second filter on the bioprocess container. This will allow you to have another source and to mitigate any risk of those sources. In addition to supplying, you know, kind of a standard component library that's been qualified that you can choose from, the supplier can also provide you with a component information summary. This will give you the understanding of the material makeup within each one of those components and so forth. This will help in the L&E and biologic testing process that happens with the end user. So when looking at all of these different connectors and fittings, we do go through a validation process with these connectors. There are a number of them. Some of the things that we do to make sure that the connectors are integral. Anytime we're validating a new component, we look at leak rate, pressure, hold, time testing, and then we also look at the temperature range in which those components and tubings can hold up. So there is a full validation of any of the components that we're providing on the bioprocess container. Those reports can be provided as well to help you with the selection process of those various components that are being used. So now that you know, we've talked about the film choice, the components, there are various designs throughout the entire upstream and downstream process. The recommendation again would be to look at this from a holistic approach. Design the container that meets that specific application. So there, you may end up with, you know, 100 SKUs, From there, what we do is we go through a process with the customer to reduce costs and design. So there may be some designs that, for example, a mixing bag, you may be using it for media hydration, buffer prep, cooling and storage for the downstream process, viral inactivation, harvest hold. Design it to that application, and then we can narrow that down to the SKUs that may cover, those various processes. So if there's a powder port, that can be closed off. For downstream, you may have sensors and so forth and other mixing bags that are not utilized, but we can, plug those off, and at least you have the same base SKU number across the mixing platforms to lower your SKU creation and management. This also helps, obviously, from a quality control perspective. Quality within your facilities can look at narrowing down the SKU numbers that they have to review and qualify. In addition, from a supplier standpoint, this means lot sizes go up, and we're able to forecast, look at those lot sizes, and reduce the amount of waste that goes through the production process. So by reducing the number of SKUs, we should have higher lot sizes, which overall improves the costing back to the end user. And then last, if you do have a critical process step that you want to utilize a certain SKU number, there are line sets that we can build that we can attach to allow you to stay with that final SKU without having to design a new product for that certain application. All right, and then in regards to the process optimization, there's ways that suppliers can help with process optimization. Some of those things, services can be provided from a feasibility process optimization, scale up in your bioreactor from a seed train perspective. And then we touched on, the BPC design and study to minimize the number of SKUs and costs. So engage the supplier closely on those various activities to help reduce the number of equipment and bioprocess containers you may have within your process. In terms of space constraints, clean room and cold room space, comes at a premium price for end users, so it's important that we maximize that space. Consult with the supplier. We can provide facility layouts to optimize the space through custom containers. We can also drop in 3D models of the equipment within your facility to show conceptual design. So, this is an example of where a customer had an issue with their cold room space, where they had a number of drums and barrels within that cold room. We went through and provided them a custom racking system with 3 SmartTainer 500-liter bins stacked upon one another in various dimensions, and really what it did was allow them to increase their cold room space by 2 to 3 times what they had before. So consult with the supplier when it comes to facility layout and space constraints. Again, in terms of scale up, there's ways, from a seed train and a reactor perspective, there may be technologies that the supplier has available to you as an end user that you may not be aware of. We can go through some of that process development and scale up work for you. In this case, a customer could potentially remove one of the bioreactors from their process, scaling from a 50-liter all the way up to a 1,000-liter, utilizing the 5-to-1 turndown ratio, including a cross-flow sparge that allows for cell growth at lower ranges within the bioreactor. So again, process optimization, have those discussions with the supplier to reduce equipment needs. So just going to touch on, integrity testing and what we can do from a supplier standpoint. If you're in a critical product hold situation where you want to look at helium testing and have your final product tested before it's shipped, suppliers have the ability to do a helium test on the equipment that allows you to look at acceptable levels of gas permeation all the way down to 3 microns. So this will help ensure that the bag is integral and that there's no leaks prior to shipping. Once the product is shipped, we also offer, point of pressure decay testing. So when you're ready to run the bioprocess container in an application, the pressure decay equipment can be used for, gross leak testing, which can point out whether there was any damage during the shipping or handling of the product into the clean room before it's used within your process. And last, it's really important that, you have, a support team, a project team that is outlined between the end user and the supplier. Those support teams would include, a dedicated project manager, individuals that have expertise in mechanical engineering, controls, polymers, and so forth that can help support those project needs. Typically, once the supplier has been selected, we do schedule a kickoff meeting with our end user. We go through schedules and critical paths and any risks that may be involved during the project. And then, again, it's critical to have a process and a plan in place with various activities that are planned during that. Map out that process in terms of schedules, documents, design, specification approvals with the right teams involved. So, the more an end user plans on this facility design from an automation layer through equipment, consumable designs, engage them often, early, have an open mind on the scope of work. We can go through and identify cost savings, optimize your process, look at the supplier's background and what kind of facilities that they've designed and helped with the overall concept of bringing consumables into the space. So I appreciate everybody's time on the call.
Now we will begin the question and answer segment of the webinar. We only have 7 minutes left, so I'm not going to be able to ask all of the questions. So I hereby inform our audience that we can email the answers to their questions. So I think the first one is for Adam. What is the greatest challenge you've had with your company embracing SUT technology from a technical and management standpoint?
That's a good question. I think I would definitely tell you the kind of joke I was making about the double-edged sword is definitely the flexibility of single use. And so technicians, supervisors, local managers at local sites specifying new products, tubing sets, new SKUs at the local level, and thus not knowing about it at the global level, and trying to integrate and coordinate the onboarding of single-use assemblies globally has been a challenge. So I think really having all of the sites acknowledge a single decision-making body so that we could coordinate our implementation of single-use technologies is definitely our biggest challenge.
Thank you, Adam. The next question, and this is the final question, unfortunately, because we are running out of time. Can you quantify the benefit of smart factory installation, and what are the main benefits that customers see?
Yeah, so that's a good question as well. From a smart factory standpoint, from there you can standardize on digital communication with your pumps, your sensors, and so forth that's used across the platforms. Along with that, really overlaying a control and design interface that allows consistent messaging across the various platforms. This will ease the burden on operator training. There's softwares out there that can overlay from a true bio perspective that allows for that same user interface, including sending that up to the MES layer. You can record maintenance schedules with your production and understand that scheduling. You can look at whether operators have been trained and overall secured network across various platforms within the operation steps.
Thank you so much, Jacob. Thank you for the webinar, Mr. Adam Goldstein and Mr. Jacob McNeil. Unfortunately, that's all the time we have left. I would like to take this opportunity to thank you and for sharing your knowledge with us. Audiences can view this webinar on demand by visiting biopharma-asia.com, and I would like to inform our viewers that Biopharma Asia will have planned webinars on topics similar to this. So again, please visit biopharma-asia.com. Thank you, everyone.
Thank you.